All right. Good afternoon, everyone. We still have some people joining in, but I'll go ahead and get started since it's 1 o'clock on the dot. Good afternoon. We're glad to have you all today for today's webinar on understanding New World Screwworm, also referred to as NWS, biology, risks, and response, featuring Dr. Edwin Burgess and Dr. Nargiza Ludgate. So this session brings together experts from both the University of Florida and the Southeastern Coastal Center for Agricultural Health and Safety. And it's going to cover the biology, history, and ongoing research surrounding this parasite and why it matters for producers, veterinarians, and public health professionals. So we're going to begin with Dr. Edwin Burgess. He specializes in veterinary entomology, and his work centers on improving control of arthropods that harm non-human vertebrates, particularly in animal production. Today, he will provide a comprehensive overview on New World Screwworm as an issue, and then we'll lead into Dr. Ludgate's brief presentation on her upcoming project on um, scenario modeling with NWS. So we have a big informative session, so I'll go ahead and pass it over to Dr. Burgess.
All right, thank you. Just gonna share my screen here real quick. All righty. Um, Today I'm gonna cover some general biology, some history, and some management considerations for the New World screwworm. Um, this has been in the news a lot. We've seen a lot. A lot of the big news outlets have started to cover this. There are some large grants coming out on this subject, and it's for a very good reason. This is a tremendously historically and currently impactful parasite, both to animals and to humans. I'm going to warn you that there are some sensitive images that are in this moving forward through the slides. So if you're squeamish, just know that there will be some images. This is not a kind and friendly fly by any means. What is New World screwworm? It is a non-biting fly whose parasitic larvae feed on the live tissues of warm-blooded animals. The adult fly is primarily a nectar feeder. The females will feed on the open wounds where she lays her eggs. But primarily, they are found in the environment, feeding on sugar resources. Very heavily component of that is plant-derived resources. There are some names that you might have encountered for this. The primary screwworm is another name. The cattle borer worm, and its Spanish translations are very commonly used to describe this fly. Cochliomeia homnivorax is its scientific name, and it is different from the Old World screwworm, which is another fly species that is found in the Old World, Africa, um, Middle East, and so on. Chrysomya beziana, which does the same kind of things that the New World screwworm does. Its larvae feed on living tissue. We do not have the Old World screwworm here in the Americas. And just an overall arching thing as of this morning, the New World screwworm also is not currently in the United States. So it is a fly that has four life stages. We call this a holometabolous insect. So it has four distinct life stages. Um, the adult, the adults lay eggs. Those eggs turn into larvae. The larvae have what are called three instars, which are essentially stages that they get bigger and bigger in. In the third instar, um, they climb out of the wound and then they fall into the soil and they pupate and they turn into these little reddish brown pill-like shapes. These are immobile, and this is where the larvae metamorphosizes into the adult fly, and then they emerge from these puparia. And the New World screwworm gets its name, the screwworm, because it looks like a screw. This picture in the upper right corner here is an example of it. And I have positioned this picture very strategically because they do feed head down with their posterior up, and they actually breathe through their posterior through these things called spiracles, which are just tubes that allow for oxygen from the environment to kind of passively perfuse through their body, and that's how they get oxygen. So they don't have lungs, they don't breathe like mammals do. And so they have to feed head down and they have to feed with their posterior up so that they can breathe. And around the edges of the margins of its body are these hairs, these very sharp pectin hairs. And what they use that for is to essentially anchor themselves. A larvae squirms, they contract and then expand, and that's how they move. And these allow them to move through that kind of liquidy, slippery tissue. And it gives it the look like a screw, hence the name the screwworm. So the life cycle is another really important thing epidemiologically that we have to care about. This is what causes this organism to be so dangerous, and it's what makes control very, very tricky with it too. So the adult is about 3 to 5 times the size of a housefly. So you've seen shiny green flies, sometimes we call them blowflies, in your backyard or out by a dumpster or something like that. They're about the same exact size as those. And the adults will live somewhere between 7 and 10 days. The eggs are about 10% the size of a dry white rice grain. And a female will lay about 350 of them at a time, and they can hatch within 24 hours. Usually, it's within a few hours of laying them that the eggs will hatch. And then the larvae will develop through those three larval instars that I talked about. They start with these really, really tiny worms. You can barely see them. And they get up to almost 2/3 of an inch in length by the time they're in that third instar. And then they will crawl out of that wound as they're kind of feeding on the live tissue in around 5 to 7 days. They turn into that reddish brown puparia, which is about the size of a tic tac breath mint. And then they pupate under the soil. So you don't actually see all these red pupae all over the ground in an area where there's been an active screwworm infestation. They're really good at hiding. And then those take about 7 to 10 days to develop. It's very temperature dependent. And that life cycle can take around 18 days. In hotter climates, it can be down to 12, 13 days. It can be very short period of time. Um, Another really important thing about this fly is that they are a very thermally sensitive fly. The reason we don't historically see this fly all over the United States in temperate states or states where there are 4 seasons is because this fly has a very tropical temperature range that it can survive in. So when the temperatures get down into the low 50s to high 40s, those puparia will actually die, and the adults will actually die as well. Or they will go into an area where they just kind of rest in a warmer area. And a lot of insects can do that. So that's one reason why we don't see this massive spread all across the United States historically, is because they are known as a very tropical fly in terms of their thermal tolerance. Another really important thing about the life cycle is that females will only mate once in their lifetime. And this is really important when I explain about the sterile insect technique a little bit later. This discovery is what led us to developing and succeeding with the sterile insect technique and why it is still considered the main tool for eradication, and what they're currently putting a lot of money and effort into is to build up this sterile insect technique capabilities. And this is some interesting pictures here that I like. They actually drop these out of planes. They currently do this. This is how they did it historically. That's the picture on the left. The picture on the right is a rearing facility where they mass rear these flies. The rearing media is extraordinarily fragrant, we will say. People who are working in this, they're definitely troopers when it comes to the horrific smell that is involved with this with rearing this and doing sterile insect technique. Identification of the fly is very difficult to do. The main thing is the adults are not evident on animals. They are not all over people. They're not like a horn fly. Those of you who deal with livestock, these flies are not going to look like or present like horn flies. There's not going to be hundreds and thousands of flies all over the animals at all times. They are scattered throughout the environment. They hide, they wait until an opportunity arises where there is an available wound. The female is very good at locating those. And so surveillance is done with traps that mimic those odors to bring them in. And so surveillance on these is very tricky too. Even with these really nicely tuned traps for doing this, of several thousand flies that you might catch in a trap, shiny green flies you might catch in a trap, you might get anywhere from 5 to a couple dozen screwworms in a batch of thousands of other flies. So it's not an incredibly dense fly in terms of populations. Um, Larval ID can be very, very tricky. So just pulling them out of a wound is extremely tricky to do. The main thing is that they're spiracles. Remember, I mentioned that earlier, where they breathe out of their posterior. This image on the left is actually a picture of that. The image on the far left of the combined image on the left is the posterior end, and you see that little black shape. That is their spiracles inside of them. And when you actually pull those out, the image on the right kind of shows the color of those spiracles. That's one way you can tell a larvae from other similar species of blowflies is they have those really dark spiracles. The other really tricky thing is the adults, right? Doing surveillance on a living animal. By the time you see it, it's already kind of too late. It's the infestation is active. So they do the surveillance on the adults. The adults are also kind of tricky to do identification on. Kind of the common one you hear is that they have these three stripes on their body, the main part of their thorax, which is where this arrow is pointing to in this image. And a closely related species is Cochleomya macellaria, which is the secondary screwworm, and does not feed on living tissue. That feeds on garbage and roadkill and dead organic matter. And the New World screwworm, actually, the middle stripe does not come all the way up to the back of its head, whereas in Cochleomya macellaria it does. The problem with this identification feature is that when the fly is dead and sitting in a trap, those stripes can fade pretty quickly. So it becomes somewhat unreliable. The other problem is that the fly looks remarkably like 93 other species of blowflies that are in the United States. Same kind of blue-green, shiny, metallic body, the same size. Many of them have orange heads. This image on the far left here, this is the screwworm on the top, and this is Cochleomya macellaria on the bottom. One of the more waterproof ways to do adult identification is under a microscope. In this image on the right here, you can see this, that the Cochleomya macellaria, the secondary screwworm, does not have these hairs in between its eyes and kind of the midline or the midline of its head, whereas New World screwworm do have a bunch of hairs on that side. But this requires a microscope. It requires specific training. So you can see that this is kind of a difficult thing to do surveillance on. So the biology, so progression of infestation, we call this myiasis. This is considered primary myiasis. So this is that the larvae is feeding on living tissue and it initiates the feeding. That's primary myiasis. And so what they do on the far left, you can see an image here where there's a bunch of the female flies on a wound. They're starting to lay eggs around the margin of that wound. This middle image on the left, second from the left, is a batch of eggs that a female has laid. You can see some active larvae on the inside of that wound as well. Um, and then the larvae will start to eat that living tissue, and then mortality often results from this, because unlike other flies, which will feed primarily on dead tissue on an animal, or they're sometimes used for wound debridement in human medical practice. Those types of flies are not New World screwworm flies, and they eat dead tissue around the margins of a wound. The problem with New World screwworm is that it eats living tissue, and what it does is it keeps digging deeper and deeper and deeper into the animal. So it expands and deepens the wound, and that causes some really, really bad problems. So just some key facts here. The wounds do not have to be big. The eggs can be laid on as small as a vacated tick bite. There have been documentation of that. They do not need much room to get in and break the skin. They will lay their eggs also in mucosal membranes as well. They'll lay around the nose of an animal. The recent case that was in Florida was actually a male who had screwworm in his sinuses. That the female had laid eggs around his nose, and they got up into his sinuses. So they will do that. Infestations create this horrible smell. In cattle, when there's an infestation in a herd, you can smell it from many meters away. It smells like the most awful, deathly smell you've ever smelled in your life. Um, and I showed you that image, that one that was second to the left. There was a female laying eggs, and then there were larvae underneath. This is the big issue with this fly, is that they have these overlapping generations. So once a wound starts to expand and smell bad, it attracts other females to it to lay their eggs. And so you have late instar larvae that are getting ready to pupate, and you have young larvae that are being introduced from new females to continue progressing the wound. And so this is kind of a rapid way that these wounds can exacerbate and get really bad and ultimately cause mortality. Again, the larvae feed on live tissue only. When the animal dies, the larvae die very quickly, within half an hour to a couple of hours. As soon as the body starts to cool off, the larvae cannot survive in it because of that tight temperature tolerance that they have. With the exception of late instar larvae can exit the wound early and begin pupation. So once they get to that instar 3 stage, they usually can arrest feeding and climb out of a wound and pupate. So just because something dies that has this does not mean that all the larvae in it are going to be dead, and that's kind of a closed system. It can actually, though, some of those later aged larvae can climb out of the wound and pupate. Um, the mortality and morbidity is a result of those larvae progressing into the wound. They will infiltrate into the body cavity, into organs. When there's wounds like dehorning wounds, they can enter the skull, start eating the brain. They do not really care what live tissue they're eating. And that's what often causes mortality. Um, and a secondary thing is, because the wounds get so deep and because the larvae are kind of microbially rich, you could get these very deep systemic infections that cause septicemia, which can also cause mortality. And that mortality can happen very quickly, within a week to 2 weeks, if it's left untreated. Smaller animals it can be even faster because there's less tissue for the larvae to kind of go through and before it gets into vital organs and such. So the history of the New World screwworm. So it's been around for a very long time. It's estimated that it's been in the United States since at least 1842, so predating the Civil War. It was primarily in the southwestern United States. This map on the left here is an overwintering history. And I like this map because it kind of gives you its typical range. Again, I mentioned that it's a very tropical fly, so we don't see it making these huge migrations. These are usually seasonal when they happen, and then the populations regress back down towards essentially the warmest parts of the United States. 1951 is a good example year because we had an abnormally warm year that year, so it enabled the fly to move up into much farther northern reaches of the United States than historically it was known to. Um, and in that 1951-52 winter survival, we had a mild winter. They kind of survived in a much higher range than they normally do. That dark blue, though, is kind of the typical overwintering range that we see. And then the one on the right is kind of a timeline map of eradication, getting rid of it in the United States. So by 1966, it was completely eradicated and announced eradicated from the United States. The efforts started in Florida, which I'm going to go through in a little bit. But this map is nice because it shows you how long it took to actually push it all the way down to the Darien Gap in Panama. I mean, it was a lot of years. This is a logistically very complicated method. This sterile insect technique method it's very complicated, and it took a long time to get it pushed all the way down. So I'll give you some historical, some timeline stuff. Again, prior to 1933, this fly caused hundreds of millions of dollars in damage to the animal agriculture and wildlife across the United States. In 1933, it was introduced to Florida through the migration of infested animals. Um, so this was not something that the fly just naturally moved. And I always try to mention this, this fly existed in the United States since before the Civil War. It wasn't until 1933 that it moved to Florida. And why I mention that as it's very important because it shows how important the movement of infested animals are in moving these populations because these flies are very thermally restricted. It does not allow them the time to move across the United States before seasons kind of kick in, and it makes them, biologically or thermally unable to kind of complete the trip across the United States. And so by 1957, eradication was initiated in Florida. It was successful. The Florida Cattlemen's Association was a huge reason that all the SIT stuff we see now is implemented because of a grassroots movement that they had to raise money from cattle ranchers in the state of Florida to essentially invest in this. And the US government saw how this worked and matched the funds. And that's what ultimately enabled the first successful eradication, which happened in Florida. That happened by 1959, and then I had mentioned by 1966 it was announced eradicated from the United States. So the current status, there's a really great website, screwworm.gov, has all this really great information right now. These updated maps that they update every Tuesday. Um, this is a map as of March 19th, 2026. So this is the most recent map that's available right now. And the red dots are the ones you want to kind of train your eye to. Those are the current active cases as of March 19th. The closest one being in Tamaulipas, which is about 230 miles from the southern U.S. border. And there are 34 active cases there right now, mainly in animals. And just to kind of give you an overview too, this does affect humans as well. We always talk about livestock, but this is important to humans as well. In 2025, throughout the Americas, there were 1,000 human cases that were reported. 92 were in Mexico, and there were 5 related fatalities to those infestations in Mexico with humans. So I'm going to talk about the sterile insect technique a little bit here. So the timeline on this, it's a really cool story if you're kind of a science nerd. So in the 1930s, the New World Screwworm expanded across the United States, and they used some very old school things like benzol and pine tar oil, an old product called Smear 62, and trapping. And also just manual removal of the larvae. I've talked to several old timers in the state of Florida who, have worked on cattle ranch for their entire life. And when they were kids, they used to get these forceps, and their parents would send them out to their animals, and they would treat them, and then they would manually go around the animals and pick out all the larvae from the wounds. 1933, the New World Screwworm was identified as a unique species. Before this, they just thought it was kind of all blowflies that did this. So that was a landmark discovery. And that's what really kicked off the biological study of the fly as a species. In 1946, it was notable that Hermann Mueller, he won the Nobel Prize for radiation sterilization of fruit flies. And this is what got Edward Nippling, who was in charge of this SIT efforts with the USDA, got him interested in this as a potential way of controlling New World Screwworm. And if you go on, there's a really great USDA website that has all these historical documents archived. You can read their letters back and forth to each other. It's some really, really cool stuff to read if you're into that sort of thing. And so 1951 to 53, the radiation sterilized New World Screwworm were tested on Sanibel Island in Florida from an Orlando facility that was built to rear the flies, but it did not eradicate the screwworm. And there's a good reason for that. If you know anything about the geography of Florida, Sanibel Island is only a few miles off the coast. And so when they were releasing the flies, the sterile flies on the island, what they were finding was flies were moving across back onto the island. And so it was not a geographically isolated enough testing ground to prove that this would work. And so then they moved to Curacao, which is a much more distant island, and they retried this, and it ended up working. And so in 1957, they raised all their money in the state with the Cattlemen's Association. Um, and there was a couple of facilities in the Orlando area that produced about 100 million sterile flies per week. That is the type of numbers we're talking about to make this type of technique work. In 1958, we had an extremely cold winter in Florida. You saw that historical map where the average overwintering didn't really go north of Orlando. In this year, the populations that were overwintering were pretty much isolated to Miami-Dade County. So it had kind of trapped them into one geographical area. And so they concentrated their efforts on that area. And that's what ended up resulting in such a quick eradication in the state of Florida was a really cold winter. And then within a year, that was done because of that. And the rest of the US saw how good this worked. US government kicked in all of their money and resources for this, and that's what led to the eradication. Some other things to consider here are the management. Besides sterile insect technique, right now we're in a stage where there's about 2 years before we're going to be fully built up to be able to be pushing this back. It takes a long time to build the facilities. It takes a long time to rear that number of flies, to hire and train people. So what we have right now is surveillance, and we have insecticidal management tools available. And so that's really key right now, and that's going to hold the fort down until the SIT efforts really kick up to the point that we need them at. So prevention is going to be wound mitigation. It's going to be that surveillance. That's the other thing about screwworm. When you get an infestation, it is extremely painful. These larvae are eating live tissue, right? So it is a very, very painful thing when you have it. Animals that have it are showing extreme signs of stress when they have it. They're not eating. All the different types of, clinical signs that they give that they're under extreme stress and pain is very evident with them. It's also that really foul smell that you get from it. You can smell it from a long distance, and that usually indicates it pretty well. If you suspect it, you contact your veterinarian, you contact a health professional. You do not move the larvae from it. You don't transport animals, nothing like that. It needs to move because remember, those larvae crawl out, burrow under the soil. Impossible to find them at that point. You don't treat it yourself. You need to have medical or veterinary support to do that. And then contacting FDACS, USDA, APHIS, those related authorities is a top priority. We do have some chemical interventions. I have some up here that are geared towards animals right now. So there are quite a few insecticides that we have available for animal treatment. I know they're working through some of this stuff with humans right now with the FDA. If you want any further information, this QR code is really good for kind of looking at what the USDA is recommending for this stuff right now. And that is it for me for right now. If there are any questions later, happy to answer them.
Great. Thank you so much for sharing, Ted. And again, if you have any questions, please put them in the Q&A feature, and at the end, we'll have the speakers go through them. So next up is Dr. Nargiza Ludgate. She's a research assistant scientist with the Southeastern Coastal Center for Agricultural Health and Safety. She has an up-and-coming project that's still in the works, and we'll provide an update on that at a later date. But for now, she's going to go ahead and introduce it related to New World Screwworm. I'm going to share her slides for her, and she will present. So you can go ahead and, let me see. Can you see that, okay,
Thank you.
Lisa?
Yes, I can see them very well. Thank you. Thank you very much for this opportunity to present our project titled Modeling New World Screwworm Infestation Scenarios and the Economic Impacts on Florida's Livestock Industry. This grant is funded by CDC's National Institute for Occupational Safety and Health. Next slide, please. This is our team. It is very interdisciplinary. I bring ecological and economic evaluation lens, while my colleague, Dr. Guru Chu, who is a co-PI, specializes in agricultural safety and health. And our colleague, Hannah Baker, is an economist from Range Cattle Research and Education Center in Ona. Through this process, we are consulting with Ted and other colleagues in the entomology department for disease dynamic and risk assessment. And we're in the process of getting a student who will help us with the modeling work. Next slide. Now, what motivated us to pursue this project? Livestock industry is very important for Florida's agricultural economy. Florida is home to 1.56 million cattle and about 385,000 horses. And together they bring or generate over 8.3 billion in annual revenue. But the value of this industry is not only in dollars. It provides thousands of jobs, keeps local supply chains running, and even contributes to state's cultural heritage. Yet recently, the industry has faced several challenges, especially driven by the weather changes. Hurricanes, floods, intense heat waves have taken a toll on the herds. They lowered productivity as well as disrupted some of the operations. These extreme weather events have made Florida's livestock sector more vulnerable than ever. To protect Florida's livestock industry, there is a growing need for smarter management practices, stronger infrastructure, and targeted support, ensuring that Florida's livestock can continue to thrive for the communities and for the people who depend on it. More recently, the New World Screwworm has passed the longstanding Darien Gap barrier in Panama that Ted just presented, and surging northward through Central America and Mexico. The flies have reached the northern Mexico and causing USDA to suspend live animal imports to protect US livestock industry. Florida, due to its subtropical climate, changing weather patterns, high livestock density, and extensive trade, tourism, and wildlife movement, has a heightened risk. This all increases the likelihood of introduction and spread of the New World Screwworm. And the reemergence of New World Screwworm could therefore result in substantial economic, biological, and ecological impacts on the state. Next slide, please. So with this in mind, We propose to model new World Screwworm infestation scenarios and assess their economic impact on Florida's livestock industry. Our specific research objectives include quantify direct and indirect economic impacts of hypothetical outbreaks of varying scale and duration on Florida's cattle and equine sectors, analyze key outbreak drivers, including climate variability, livestock density, animal movement patterns, to understand how these factors influence the severity, speed, and economic costs of infestations. We also want to evaluate the costs and benefits of a range of mitigation strategies, including sterile insect technique deployment, quarantine measures, veterinary treatment protocols, along with the public awareness campaigns. We also want to assess the market level implications of trade disruptions and regulatory responses on producer incomes and supply chain continuity. We will also put effort to identify cascading effects on rural economies, including impacts on labor markets, input suppliers, veterinary services, and the feed industry. And finally, Dr. Guru Chu, who specializes in agricultural safety and health risks, will examine the occupational safety, who specializes in agricultural safety and health, will examine these risks on Florida ranchers, farmers, ranch and farm workers, and other industry stakeholders. Next slide, please. To meet these objectives, we are developing a multi-layered modeling framework that brings together risk assessment, disease dynamics, and economic analysis in one integrated approach. First, we will apply special risk models to identify areas in Florida that are most vulnerable to screwworm establishment. These models will combine data on weather conditions, livestock density, wildlife presence, and potential entry points such as ports and high traffic animal movement corridors to identify geographic hotspots where surveillance and prevention efforts could be prioritized. And please remember, these are all hypothetical scenarios. Second, we will use stochastic outbreak simulations to better understand how screwworm infestation could spread under different conditions. By incorporating uncertainty and variability, the simulations can allow to test a range of scenarios, including delayed detection, varying levels of response capacity, and different control strategies. We hope that scenario modeling will help assess how quickly an outbreak could escalate and identify effective points for intervention. Third, we're estimating economic impact of potential outbreaks under different scenarios. These analysis will go beyond direct livestock losses, such as mortality, reduced productivity, and treatment costs, to also capture broader ripple effects across markets. This includes disruptions to supply chains, price fluctuations, trade restrictions, and impacts on related industries that depend on livestock production. And finally, we're going to conduct cost-benefit analysis to compare alternative strategies and determine which approaches provide a good return on investment. These strategies include early detection and surveillance systems, SIT program, community outreach, and producer engagement. The latter draws on the lessons learned from Florida's successful response in 2016-17 screwworm outbreak in the Florida Keys, where coordinated efforts, including public awareness and rapid intervention, were very critical to eradicating the infection among key deer and preventing wider spread. Hopefully, the findings of this modeling work could provide decision makers with actionable evidence-based insights to strengthen preparedness and response capacity. Next slide, please. To ensure that the research is grounded in industry realities, we are working with the Florida's livestock stakeholders to conduct interviews and capture local knowledge, risk perceptions, and preferences regarding potential new world screwworm mitigation strategies. Collect some farm-level economic data from cattle and equine producers across the state through surveys, and working in coordination with the Florida's Cattlemen Association and the University of Florida IFAS's task force on New World Screwworm. I think this is all. The next slide is my contact information. Thank you.
All right, thank you so much, Nargiza. So it looks like we don't have any questions in the Q&A. So if you're still in the participants, go ahead and drop your questions if you have any. We'll stick around for a few more minutes, but if not, thank you so much for attending, and thank you both, Dr. Burgess and Dr. Ludgate, for presenting today. All right, looks like no questions, so I will go ahead and stop recording.